This is the second lecture for Friday, April 17th. Just to remind you, we're looking at different marine ecosystems. We've gone through these different coastal ones. We looked at the open ocean, huge segments of the ocean. We're also looking at this other very large portion of the ocean, an ecosystem that's characterized by relatively stable conditions as opposed to being so dynamic as you find in all those coastal ecosystems, and quite a bit the same in the ocean, open ocean, oceanic ecosystems. So this is a very static type of ecosystem. There are some changes, but there's not much of a seasonal change. You don't have a bunch of organisms that are moving all over the place. So it differs from these other ecosystems in that respect. We were looking at one group of animals that I think is really interesting, the deep sea sharks, and how they've adapted to different environmental conditions in the deep sea. There are things that are certainly challenges for a lot of marine organisms, but this low biomass and the reduced amount of energy, the reduced amount of food for a lot of deep sea organisms is certainly a big challenge, and one that's met oftentimes, quite commonly, by organisms achieving neutral buoyancy. And so we looked at the prototypical shark, deep sea shark, and saw that for the most part they have a large liver, they have a lot of oil in their liver, and that liver contains these low density lipid classes. And where we left off in the other lecture was that there are differences within groups of these deep sea sharks, and they're certainly not all the same. So I want to look at differences in species amongst these different species. There's different ways that they attain neutral buoyancy. There's differences between males and females, and there's differences between small sharks and large sharks. So that's what we'll spend our time on in this second lecture. So first of all, if you look at the typical deep sea shark, first look at this figure on the right. And you see these yellow dots. Those are different species of deep sea sharks. That dashed red line is neutral buoyancy, a density of 0. So you're neither heavier or lighter than the water that surrounds you. And then you move up in the red and the blue and those circles and squares for shallower water sharks, and you get up to the point where, like most bony fish, the bodies of sharks, typical sharks, besides deep sea ones, are about 5% heavier than the water that they're in. For bony fish, they make up that 5% difference with their swim bladders. For sharks, they don't have swim bladders. They make up that difference, one, with their liver, so hydrostatic mechanisms, lift, and two, with their fins and their tails by moving through the water column through hydrodynamic mechanisms. Now, if you don't want to be up in the water column, then it's good to be more dense than the seawater. And this 5% greater density is a good thing because you lay on the bottom. The figure on the left shows within that red rectangle a number of different species of deep water sharks. Now, these are the typical deep water sharks that we were looking at before. They've got a big liver, they've got a lot of oil, and in that oil they've got a lot of squalene, which is the yellow in those columns, and they have a certain amount of diacylglycerol ether, which is the magenta colored. And what's left, the rest of the lipids that you find in their liver oil, is primarily triglyceride, that blue. So if you look at the combination of yellow and pink, yellow and magenta, in that red rectangle, you'll see that for all those sharks that are in there, the combination of those two is at least 80% of their liver oil, or very close to it. In some, it's 90%, and one of them, it's almost 100%. So that's the typical deep water shark. A lot of squalene, that yellow, and made up the rest of the liver oil is almost all diacylglycerol ether. Both of those are low density lipids, much less dense than the blue triglyceride, so they provide a lot more lift. Their oil is very buoyant, their livers are very buoyant, And the result is that the body of these sharks, the whole body, is neutrally buoyant. But if you look at the left of that red rectangle where it says 6-gill, now that's all diacylglycerol ether and triglyceride. No yellow, no squalene. If you look to the right of that, there's this PLS. That's a plunket shark that looks very similar. It's closely related to all those sharks in that red rectangle. That plunket shark is outwardly very similar. You'd have a hard time telling it apart from these other species. But if you looked at its liver oil, there's a huge difference because there's only trace amounts of squalene, but there's a huge amount of diacylglycerol ether. So very closely related species phylogenetically, but biochemically very different. And then there's spiny dogfish, which is kind of an intermediate. It's not really a lot of places it's shallow, but it can go into relatively deep water. For the most part, it's a shallow water shark, though. But it's closely related to all these species. And you notice it has a little diacylglycerol ether. And then these APR E and C and A, those are cat sharks that are not very closely related at all to those in that red rectangle. They're at the opposite end evolutionarily, phylogenetically, and you'll notice that there's quite a bit of difference. One of them has some squalene and some diacylglycerol ether. One of them has some diacylglycerol ether similar to a spiny dogfish. One of them just all triglyceride, which is similar to a sandbar shark, except the sandbar shark shows that typical distribution of lipid classes that you find in shallow water organisms, and a lot of organisms, in fact, with almost all triglyceride, but some free fatty acids that are these chains that come from phospholipids that are in their cell membranes for the most part. So you see, in the figure on the right shows that all these different species are close to neutral buoyancy, maybe a little above, a little below, but they're pretty close to neutral buoyancy. And the figure on the left that shows the lipid composition shows that this one group in the red rectangle, pretty similar for the most part. And this is what people thought for the longest time. That's what deep sea sharks do. Big liver, a lot of squalene, a lot of diacylglycerol ether. But you see, there's quite a bit of variability amongst the species. So let's look at these different species, and we'll see they all achieve neutral buoyancy. However, they do it different ways. So we're going to look at 3 different species or groups of species. The 6-gill shark, which is this big shark that's not so closely related to all those with the yellow and the magenta. The plunket shark, which is very similar to these, very closely related. And then the deep water cat sharks, or cat sharks, in the genus Aprosturus. All 3 of those groups, or different species, attain neutral buoyancy, but they do it in different ways. So we're going to look at their size of their liver. We're going to look at the amount of oil in their liver. Those are 2 characteristics that are common in deep water sharks, 2 things that are associated with neutral buoyancy in most deep water sharks. And then we'll look at the content of their muscle, not just their liver, but their muscle as well. Because we'll see that it's not just about the liver in achieving neutral buoyancy, but it's also about their muscle. So if you look at the density of the liver oil in this figure on the right, you see that the plunket shark and the 6-gill shark in that green rectangle, they have liver oil that's has a density of about 0.9, which is kind of what you'd expect with a combination of either diacylglycerol ether or diacylglycerol ether and triglyceride. So, in fact, if we expand this green rectangle over here to the Aprosturus species as well, then you see the density of the liver lipid for the Aprosturus are quite a bit higher. Some of them have little squalene, but most of them don't. If you look at back here in the figure on the left, you see that one of them, Apistris E, has some squalene and a little diacylglycerol ether. Apistris C has some diacylglycerol ether, but all three of those species are mostly triglyceride, so you'd expect this higher liver oil density. But they all are pretty close to neutral buoyancy, even though their liver oil isn't what is doing it for them. So let's look at the six-gill shark first of all. Six-gill shark doesn't have any squalene. In fact, these sharks, all those yellow and the plunket shark, those are squaloid sharks. That's with a name. They're in the family Squalidae, or they're in the order Squaliformes, which is where squalene comes from in the first place. Spiny dogfish is also in that, so is the plunket shark. But the six-gill shark, it's got a lot of diacylglycerol ether. So it's, and when we looked at its liver oil was more dense, still pretty light, but it's more dense than all those yellow deep water sharks. So hardly any squalene, a lot of diacylglycerol ether. So their oil doesn't provide as much buoyancy, and yet six-gill sharks are neutrally buoyant. How do they do that? Well, one of the things that six-gill sharks have, as far as a characteristic, is a very large amount of oil in their liver. So their oil might not be as dense, as buoyant as some of these other species, but the amount of oil that they have is one of the highest amongst all these different deep-sea sharks that have been examined. Their oil, their liver is really, really oily. So the oil might not be as buoyant, but they have more of it. And there's more buoyant oil than the whatever else makes up the liver, which is more dense tissue. Another thing is, and this really makes a difference, is if you look in the muscle of six-gill sharks, not just six-gill sharks, but another closely related species, the sleeper shark. It's not that closely related, but it's similar in the fact that both of these are really big. Six gills can be 14 feet long. Sleeper sharks, 20 feet long. Big sharks, whereas all these other ones are relatively small. If they're 5 feet, that's a giant. But the six-gill and the sleeper shark are 14, 15, 20 feet long. If you look at the muscle, and you look at the amount of lipid or oil in their muscle, these two big sharks are very unusual in the fact that both of them have a lot of oil in their muscle. Their muscle is almost 20% lipid. And not only that, but the lipid of the sleeper sharks contains diacylglycerol ether. This is muscle in the lipid. 20% lipid in the muscle, and some of that lipid is at low density lipid classes. If you look at these other typical deep water sharks that have these big oily livers with a lot of low density lipids, they don't have very much oil in their muscle at all. 2 or 3%, which is more along the lines of what you find in a lot of animals. Only 2 or 3%, so there's 10 times as much lipid in the muscle of sleeper sharks. And with six-gill sharks, you've got a very different lipid composition. You've got triglycerides. The sleeper shark has a lot of triglycerides and diacylglycerol ether, whereas most of these, the muscles in the typical deep water sharks have lipid classes that are dominated by phospholipids. There's a lot of cells, and there's a lot of protein, but there's a lot of cells in muscle, and there's a lot of cell membranes, a lot of phospholipids that make up the cell membrane. So six-gill sharks in particular, sleeper sharks are similar, they might not have the squalene, they might not even have that much diacylglycerol ether, but they have a very oily liver, and they can do a very good job of making that up with their muscle. So in the end, you've got six-gill sharks and sleeper sharks. They've been observed in underwater videos very close to neutral buoyancy, swimming along really, really, really slowly because they're really floating. Now, what about the plunket shark? This is one that's very similar, very hard to distinguish between these other species that are very low density. Well, the plunket shark liver, remember, doesn't have any squalene. It's got a lot of diacylglycerol ether, so the oil is less dense than a typical shark, but it's not as buoyant as all these other deep water sharks that have squalene. They have little squalene, little if any squalene. So what do they do to make up this difference of not having squalene that's contributing to their buoyancy and making their liver oil very low density? Well, one of the characteristics of these plunket sharks is of all these sharks that have been examined, they have by far the biggest liver. They've got a huge liver. It's 25% of their body weight. That is very big. And in addition to having a very big liver, they have a very oily liver. If you look in the figure down on the bottom, you can see the amount of oil. It's over 80% of their liver is oil. If you look at the figure up in the top, all the way on the right, there's the plunket shark up there at 25%. A big giant liver and a very oily liver. Now that oil might not contain any squalene, and that oil might provide less lift than a more low-density lipid-rich oil of some other species that's got a lot of squalene. But still, plunket sharks make up this difference with a big, oily liver. This third group that I want to look at in terms of the differences amongst species are cat sharks in the genus Aprosteris. Now, they don't have that big of a liver, some of these, and they don't have that much oil, some of these, and one species has some squalene, but the other ones don't have any squalene or very, very small amounts, and they don't have much diacylglycerol ether as well. It's not surprising that they're very different from all these dogfish, which are those typical shallow water or deep water ones that are all these yellow dots in this figure on the left. But one thing about these cat sharks is that they have a very watery muscle. And if you look at the figure on the left, this shows the percentage of water that's in the muscle. And so typical shallow water shark, they're about 75% water, the muscle is. And you get into these deep sea sharks, they have more watery muscle. But if you look in that figure in the green rectangle, the species with the most watery muscle, 3 out of the 4 are those Aprosteris, very watery muscle. And so they're not using their liver so much for lift as they are their muscle. Just like with those big sharks, the six-gill and the sleeper shark, they're using their muscle. And this water, it might not be low-density lipid, but they're replacing something that's heavy, which is muscle, with something that's not heavy, water. And in fact, if you look at their body without their liver, so this is the body this is what's heavier than seawater, and you look down in that circle, that green circle in the figure on the right, all those, 3 of those 4 dots there are these cat sharks. So their body without their liver is not that much more dense than seawater, whereas a typical deep water shark is 2 or 3% heavier than seawater, and you get into shallow water sharks and it's that 5% heavier that I mentioned before. So that is what these cat sharks do. They increase their buoyancy by storing water and have very watery muscle. that reduces their density overall, not with their liver, but with their muscle. So those are the differences you see amongst the species. That's just some examples of the variability you find within and among species. So I'm going to stop there, and on Monday we can take up this topic again, looking at differences between males and females, and looking at some of the differences in different size deep water sharks.